All right, let's get started. So hello, everyone. I am Lisa Rani. I'm the Director of Research at FARA. Welcome to the FARA Flash Talks. This is one of the ways we celebrate FA Awareness Month, the month of May. And today you'll hear from 5 junior investigators who will present their work, their research project on FA. And all the presenters today are being funded by FARA. We have 2 grad students working on obtaining their PhD, 2 postdoctoral fellows, also FARA fellows, and 1 scientist funded by FARA by an organization called Uplifting Athletes, who funds research in rare diseases. The format of the talk is the same as for previous years and from the same as the talks that we had 2 weeks ago. We are asking presenters to explain their research in 5 minutes and using 1 single slide. And their goal is to communicate their finding to us in a lay language. Because these can be a little challenging for scientists sometimes who are used to talk to other scientists, we can take this opportunity to ask them questions to clarify what they presented and ask them to explain what the research means and what it means for people with FA. And you can do that by typing your questions in the Q&A box. We are recording the presentations and we will post them on YouTube so you can watch them later. These are the speakers today. But before we start, let me give you a little summary of what we'll hear today. You'll hear first how we're trying to understand not only the changes that FA brings to the body, but also importantly, when these changes occur. We know that some changes in the brain of people with FA occur over time, over their lifetime, but we also suspect that some of these changes are already present when FAs are born. And Lucy is using a mouse model of FA to understand these very early changes in the nervous system and try to distinguish them from changes that occur later in life. And you can understand how these has implications for developing therapies, because it is as important to know what we want to target as it is to know when. And then you'll hear how we can use blood to try to understand and predict FA. Shweta has found signs of inflammation by analyzing blood of people with FA. So inflammation is part of the physiological way that the body tries to heal after injury. So it's not necessarily a bad thing, but when it's chronic or persistent, it can become pathological. And Shweta is trying to understand the role of inflammation in the pathology of FA and see if these changes can be used as a blood-based biomarker, a measure used to monitor disease progression and perhaps even to monitor the effect of treatments. And then the last 3 talks will be examples of research to develop therapies. So Jodi, who is a postdoctoral fellow from Stanford and could not unfortunately be here today, but sent her presentation so we can show it to you. She's studying the mitochondria, the compartment of the cells where frataxin resides and the part of the cell that is responsible to make energy. As you can imagine, this is a very important component. So it needs to be functioning properly. And when it isn't, it needs to be removed and cleared so that it can be recycled into a functioning part, like parts of a motor that can be recycled to repair a malfunctioning one. And Sujodi is testing drugs that are targeting this mitochondrial recycling system to see if they show benefit in FA. Another therapeutic avenue for FA that you probably all know is gene therapy. which consists of delivering a functioning frataxin gene to cells. And you probably all know that one of the challenges of developing gene therapy is to reach the brain. The brain is protected by a barrier called the blood-brain barrier, which is very important to protect the brain from pathogens like viruses and other harmful substances, things you eat, drugs you take, but it's also a barrier for that special vehicle that researchers use to deliver gene therapy. And what Chanfang is doing is designing in the lab a new vehicle that can cross the blood-brain barrier and deliver frataxin to the brain, like an engineer would design a new space shuttle. And finally, you'll hear how bone marrow stem cells, called hematopoietic stem cells, have the potential to treat FA. These stem cells are made in the bone marrow, but they can also be found in blood. And Anusha is trying to prove that by isolating these cells from blood of FAers, then removing the GA repeats and reintroducing these cells in the bone marrow of patients, and this is called an autologous transplantation, she can treat the disease. And today she will show how doing this in a mouse model of FA improve the FA-like defects in the mouse. Okay, so let's get started. We have two members of our community today moderating here with me, Beth Hamilton and Veronique Latt, together with Kelly Madden, FARA's patient engagement manager. And I will just turn it over to them to introduce themselves first and then the speakers.
So Elizabeth won't start. So hi, everybody. I'm Veronique Latt from the French advocacy group for FA, which is called AFAF, Association Française d'Ataxie de Friedreich. I'm one of the vice presidents. AFAF exists since 40 years, and we are a group of all patients or parents of diseased children, and we are targeting several objectives, providing information and support to patients and their family, also representing them in several groups concerning health in France, representing them through European Association and also in partnership with FARA. And we are also granting some research projects in a more modest way than FARA can do. And also we are, as a consequence, collecting funds to be able to provide those activities. And thank you for letting us speak today and introduce this new researcher.
Thank you. My name is Elizabeth Hamilton. I am a social worker. I'm a mom of a kiddo with Friedreich's ataxia who is absolutely incredible. I also have the privilege of writing for FA News. I have a column called Recalibrating. And I will also say and encourage you all to ask questions, especially if you don't have a research background. I barely passed genetics and molecular biology in college, but I am here. And so I really want to encourage you all as you listen, if there's something that you don't understand, there's no question that you can ask that will not help us all learn together.
So we introduce the first speaker, who is Lucy. She is currently investigating how FA could affect earlier development neurons. She will present our difficult to pronounce it. Sensory neurons. She will present it this research today. And so, as Elizabeth encouraged it, feel free. We will collect your question or ask our own question if some place remain.
Thank you very much. I will share my screen. Okay. You can see my screen?
Yeah, looks good.
Okay. So hello everybody, I'm Lucy Hermé. I'm a first year PhD student in the team of Hélène Spuccio in the Neuro-muscular Institute in Lyon. so in France. And I would like to thank you first, FARA, for giving me the opportunity to present my work today during this flash talk session, and also to awarding me with a PhD fellowship to allow me to do my PhD. So my PhD project aims to decipher how early does FA start impacting your body. So one of the main features of FA is the loss of proprioception. And proprioception is the ability of your body to sense its position in space. And proprioception is due to, is controlled by the proprioceptive neurons, which is a specific type of cells. And these proprioceptive neurons are formed during embryogenesis, so before birth. And where are they located? So they are located in specific structure that are positioned all along the spine in pairs, so between both sides of the spinal cord. And within this structure that are called dorsal root ganglia, also DRG, there are three main types of sensory neurons that either control pressure and touch, or the transmission of pain, and finally the proprioception, and that is controlled by the proprioceptive neurons. And all of these neurons will acquire their final form through a dynamic process during embryogenesis. So in mouse, a mouse takes 19 days to develop, and starting 10 days of development, you can already follow the cell that will become after mature proprioceptive neurons. So first you start with a neuron precursor that express specific marker, and after you will have multiple maturation step to arrive at a mature proprioceptive neuron that will express specific marker like valve, that is a really important one. So during my PhD, I'm investigating how this process is impacted in condition of low frataxin level, so in an FA context. And to do so, I'm using a newly generating mouse model that is called the frataxin D127V, and that express really low frataxin levels. So as you can see here, we are able to detect the level of frataxin, and in a mouse that doesn't have the mutation, there is high level of frataxin. So the bigger the band is, the higher the amount of frataxin there are. And in the mouse that have the mutation, you can see that the band is really thin, and so showing that the frataxin level are really low. So I'm using this mouse model to visualize the proprioceptive neurons during development. And I'm using two main microscopic techniques. So first, a 2D one, using histological cut. So on tissue sample, I'm cutting the DRG to have very thin slice of DRG. And I'm doing specific labeling of markers. So here for PIVA that I mentioned just before, that is a marker for the proprioceptive neurons. So you can see that there is a bright green spot, so that corresponds to the proprioceptive neurons. And you have the DRG that is lighted here with the dashed line. And in a control condition at E18.5, so 1 day before birth in mice, you can clearly see that there is multiple green spots, so multiple proprioceptive neurons. But in an FA context, there is a huge reduction of this green spot, so the proprioceptive neurons, showing that there is something that is happening to the proprioceptive neurons during the development, so before birth. And currently, I am developing in the lab a new technique, a new microscopic technique, to see the DRG in its total, so in 3D, and in its globality, and not on cuts. So here you have a DRG that I label with the same marker as before, so for the proprioceptive neurons. And you can see that we can spot the different type of neurons. So this is a second DRG and we can spot the different proprioceptive neurons within this DRG and see their relative distribution inside this structure. So during my PhD, I will focus on specific marker to follow this maturation process and to see how and when the system is dysregulated in presence of low frataxin levels. So thank you very much for your attention, and I'm ready to take your questions.
Thank you, Lucy. May I start? It's a very pessimistic question, but could it be that the neuron is affected from the earliest stage, so cannot even be cured?
So this is one of our hypotheses, that there is something that is going on really early on, that there is an alteration during this maturation process. So we don't know if it's not curable at all, since we don't have enough data and still it's a long way to go. But for sure, our main hypothesis is that there is something that is going on early stage.
All right. But yeah, okay. There's a question from Jennifer. I don't know, sorry, Kate. Do the people raise the question directly, or do I have to read them, please?
You can read them, yeah.
All
I think Lucy can see them, but if you read them...
All right, okay, yeah. So Lucy, if you want to answer directly to Jennifer
okay, so
question.
if the proprioceptive neurons are deficient or absent, could other type of neuron take over or perform their function? So the perform their function, no, because the proprioceptive neurons are very specific to proprioception. And within the dorsal root ganglia, there is the neurons that are present are really specific to their own function. So for the pressure and the touch, it's a specific type of neuron. For the pain, it's a specific type of neuron. And for proprioception, it's a specific type. But still, we think that there is somehow a compensation that is happening because when these mice are born, they are walking normally until a certain age. So there is something that is compensating until a certain point that we need to decipher and to investigate.
So yeah, okay. So this could be the one pist, one target for therapy, as Liz was saying at the beginning, for therapy or treatment, or.
So for therapy, I think.
What is compensating, and could it be reinforced?
Yeah, maybe, but yeah, we still need to understand how it's compensating, and because now it's still an hypothesis for us. We don't know if which type of cells are compensating, if it's neuron or not. So we still need to know that. So I don't know which question I have to start.
Do you want to pick some, Veronique, and read them, because I don't think people can see them.
All right, so Crystal, sorry if I don't pronounce the names correctly, asked if do proprioception neurons have a life cycle that is finite, determined, or do they regenerate?
So neurons in general, they have a defined time of lifetime. So once they are degenerating, they will not degenerate. They will not be like a regenerative process, unfortunately. But
so there's another question. First, a comment from Mary saying how interesting, and she's questioning. She's thanking you also and questioning what makes you interested in FA.
So yeah, it's a good question. So first, I started my master in Helen's team. Before, I didn't know what FA was, and I started my M1 and after my M2, so my master degree, doing my my internship in her team, and I discovered the disease, and I found it like the research on it was really interesting and really really a lot of passion. So yeah, it's really interesting to work on this disease. So I see that there is also a
There's one more. Yeah.
Yes, there's a question in our channel asking if you have tried multiple markers, is it possible that the proprioceptive, so neurons are there, but they are or immature or abnormal. So that's why several markers.
So I'm trying multiple marker right now. So yes, I'm combining different markers to see if these neurons are expressing like mature marker, but at the same time more immature marker. So it's still ongoing. And is it possible that the proprioceptive neurons are immature and abnormal? Yes, it's possible. It's one of our hypotheses that maybe they express mature marker, but also immature marker. So, and they are not fully functional at one point.
Lise, how long is the time for questions?
I think maybe we can move on. I don't see any more questions.
Okay.
We can.
Thank you very much.
Thank you. So we move to Shweta, who as Lise introduced, is researching if FA triggers inflammation, so and inflammatory immune response, and if yes, could it be used to be as a marker, as a biomarker for FA by detecting in blood cell, for instance? So Shantra.
Yeah. Hi. Hello, everyone. I'll just share my screen first. Is it visible?
Yep.
Okay, great. Okay, so once again, hello everyone. I am Shweta and I am conducting my doctoral research at the Department of Neurology, All India Institute of Medical Sciences in New Delhi. And my work derives as part of a collaborative team which is led by Dr. Achal Kumar Srivastav at AIMS New Delhi and Dr. Mohammad Farooq at CSIR IGIB New Delhi. And recently, I also got the FARA's Graduate Research Fellowship Award for conducting my research work. Besides, I've been manning the role of a site coordinator for FARA's Unified Study at the AIMS New Delhi. And I'm mentioning this because it is my engagement in this latter role as a coordinator and my continuous engagement with FA patients. I realized that FA as a disease is very variable and that no two patients are ever the same. So, therefore, one of the challenges for FA's clinical research program is to find out whether a new treatment would work in any particular patient or not. We know that FA primarily affects the nervous system or even heart and certain other organs, but all those organs are very difficult to get into, and it is very difficult to find out if those treatments are working in the systems I mentioned. But there is some earlier research which shows that FA triggers some sort of an inflammatory reaction in the blood. So, the driving rationale behind my research is that by studying these changes in the blood cells, we can find certain biomarkers which can help us to track and also treat FA. And my research goal is to find a way in which blood can be used as a liquid biopsy tool to assess the efficacy of new and incoming treatments in FA patients. So, towards this objective, I'm looking for certain special markers in blood which are known as biomarkers. Now, one can think of biomarkers as tiny flags which tell us about what is happening inside the body. And these biomarkers are really helpful for doctors. They can show early signs of a disease, they can help the doctors to track a disease progression. They can also tell the doctors whether a treatment is working or not. We can also compare it to, let's say, a weather forecast. And just like meteorologists use clouds and winds to predict rain, doctors can use these biomarkers to predict and monitor health conditions. And these biomarkers are of different kinds. Some can tell you whether you are healthy, others can indicate if you have a disease, and some can also tell us if a treatment is working or not. And these biomarkers can be obtained or can be found in different parts of the body. It can be in blood, it can be in urine, or it can be in any particular tissue. In our particular case, in my experiment, I'm looking for biomarkers within the blood. And we are specifically looking for biomarkers which can show if new treatments are helping us to get better. So now coming back to the slide, the left panel of my slide is a very simplistic representation of the research work that I am undertaking. And the right panel of the slide is the probabilistic outcome of this work. So going over the left side, to find these biomarkers, I'm trying to study the gene expression in the blood of FA patients. Now, to make it simpler for most of you, gene expression is like the body's instruction manual. It's your DNA telling your cells what is to be done, and by examining these instructions we can find out clues about your health. I collected blood samples from FA patients, also from certain healthy individuals for comparison. And interestingly, I have included patients which are at different stages of the disease. And this was done because I wanted to get a full picture of how FRDA develops over time. And then to find these important biomarkers, we are using a special technique which is called RNA sequencing. Now, this RNA sequencing, it's like reading the instructions which are present inside the cells. And these instructions tell the body as a whole how it is to function. And it helps us to understand the gene expression of the cells. Now, there are two ways in which the sequencing is done. The first one is the bulk cell RNA sequencing. Here, what we do is we look at the gene expression in a large group of cells, all of them taken together at a time. Now, this gives us an overall picture of what is happening inside the blood. The other one is known as the single cell RNA sequencing, wherein we look at the gene expression within individual cells. Now, we know that blood is a mix, it has different types of cells present therein. There are T cells, there are B cells, neutrophils, eosinophils, and all the other cell types. And the single cell RNA sequencing method helps us to give a detailed view of what is happening inside all those different cell types. And thereby, we can better understand the disease mechanism. It can also help us in designing new treatments and therefore find more precise biomarkers. So, I'm using a combination of both these techniques. Now, once I have sequenced the samples, I have generated all the data. Uh, I'll be analyzing this data and using bioinformatics and computational tools to find FA-specific biomarkers. Now, these biomarkers will be in the form of a group of genes whose expression levels will tell us about the disease status. Once this biomarker panel is developed, we hope it can be used in clinical trials to see if the new treatments are working or not. So it's like if you're going to participate in a clinical trial, you receive a new treatment, we will take your blood sample. We or anybody else who's going to use a biomarker panel, they can take your blood sample as a liquid biopsy. They do some PCR based tests, and using those results from the PCR based test, we can know if the treatment is working for you or not. And if it is not, then maybe the treatment can be adjusted, right? So with these biomarkers, we can personalize treatments to ensure that they are effective for you. And that's it. That's what I hope to achieve with my work, and that's all I had to say. And but before I conclude my talk, I would thank FARA for supporting my research with the graduate research fellowship. Also giving me this opportunity to present my work here. I also thank my research team and my supervisors and the patients who have consented to be a part of the study. Thank you to all of you for being a great audience, and I look forward to any questions that you have. Thank you again.
Thank you, Shweta. And thank you for the nice images that makes the scientific part more understandable.
Thank you.
Any questions?
Can I jump in and ask one, even though this isn't my time to to speak? It makes me wonder if you're able to pull this information from a blood test, if it then will help cut back on some of what we see with like the placebo effect, so that we can understand efficacy a little bit better. Because when we're doing FARs, we know that there's a little bit of a placebo effect because the person testing and the person doing it, we can see some changes, but will that kind of help cut down on that piece?
I'm sorry, I didn't really get your question. Could you?
Yeah. So when
Yeah.
we look at treatment, sometimes you can have, when you're in the placebo group, you can see that placebo effect.
Yeah,
And
true.
so when trying to understand if something really has strong efficacy, that's something we account for. So do you think this will help us understand true efficacy even better because we're cutting down on possibly what we might see placebo effect wise with other types of testing?
Oh,
Maybe that's,
yes.
yeah, okay.
Yeah, this is exactly what, biomarkers will help us do, right? They can
Right.
help, it's another measure besides, outcome measures that can be, affected by placebos. Yeah. Maybe I have a question.
Yeah.
So what you detect all this inflammation, signs of inflammation in the blood, and can you figure out from those, you were looking at the gene expression, can you figure out, what is the source of inflammation? Does it come from the brain? Does it come from the heart? Does it come from the muscle? Where does it come from?
So, although we're looking at those things yet, but one of the hypotheses is that a lot of inflammation is elicited by the DAMs, damage associated molecular patterns, which are circulating in the blood, and they come because the neurons are degenerating in Friedreich's ataxia, right? And even other neurodegenerative diseases, and that leads to the inflammatory markers occurring in the blood. Similarly, even the cardiac symptoms, when they show up, they will have certain manifestations, inflammatory manifestations, which come in the blood. So that is the source of inflammation in the blood. And we are trying to mirror back that inflammation to the site of disease pathogenesis. That is what the target is here.
There are two other questions. One from Mary saying, great talk. Can you also use those tools to prognosize, to establish the prognosis in terms of severity of the disease? And I will ask the other question after.
Yeah, definitely. That's one of the objectives, to use the set of genes as a biomarker to even assess the prognosis. Because once we know how the disease is moving, how it's moving forward, that is only when we can assess the efficacy of treatments, right? So these are both correlated.
There's another question related to your research from Heather saying, great talk. Do you think it's possible that controlling the inflammation might help slow the disease? Yeah, another question after.
The answer to that should be yes, because
Yeah.
a lot of the treatments which are working, even Vilaxon, which was approved in one way or the other, helps to bring down the inflammation. So if not directly, but then through indirect channels, inflammation might be a way to help treat the FA.
Okay, and a question from Mary saying, thank you for your work, the dedication and passion, and your passion, and asking how long have you been working with FA patients?
Oh, it is going to be, this is my 4th year actually of working with FA patients.
Sorry, I haven't heard. You said?
So this is
40?
going to be my 4th year of working with FA patients.
Okay.
It's
And
been a long engagement, and yeah.
There are other questions asking if it's, do you think it's possible that controlling this inflammation might help to slow the disease? And another, yeah, okay, let's go with this one and we take the other one after.
Again, as I said, it can, once we control the inflammation, we might slow the progression. So all these are might, because Friedreich's ataxia, it's a very complex disease. There are so many aspects to it. So it's not like one particular treatment can cure everything which is related to the disease, but then a particular treatment or a particular line of therapeutics might impact in a certain way to help slow down the progression, to make it better, to make the lives of the patients better in one way or the other.
Okay. And can this information help understand some mechanism of the disease that we
Definitely,
could not
yes.
have known? Yeah.
Yes, yes. With single cell RNA sequencing, that is what we intend to do. We are trying to understand the disease mechanisms better.
And another question is asking, is inflammation related to mitochondrial damage?
Again, the answer is yes. It is related to mitochondrial damage as well, because mitochondrial damage is the underlying is supposed to be one of the underlying phenomena for all of the FA's pathology, and that occurring in all the different tissues of the body is again bringing about inflammation.
Okay. And how will you convince other researchers to move to FA field?
um, Meet the patients. You'll be
Yeah.
convinced yourself.
Okay.
You would want to work, you would want to research on the subject.
That's a very good answer.
Yeah.
All right, shall we move on?
All right. So the third presentation will be a recorded video, since the person cannot be present today. So it's Shutropi who is investigating related oxidative stress pathway on the one implicated in Parkinson's disease. She's studying if it's also relevant to FA.
Hi everyone, my name is Sujyoti. I am a postdoc in Dr. Shinan Wang's lab at Stanford University. I would like to thank FARA for this opportunity, and I am happy to share my research with you all today. The main focus of my research is to study the cellular and molecular mechanisms of FA with an emphasis on mitochondria, or the power plant of the cells, and to identify novel mitochondrial pathways that are altered in FA patients. To study this, I use induced pluripotent stem cells, or iPSCs, that are derived from skin cells or fibroblasts of FA patients. These are further edited to generate isogenic control iPSCs, and I use these iPSCs and further differentiate them into proprioceptive sensory neurons and cardiomyocytes, which are two of the cell types most affected in FA. Here on the right is the characterization of our sensory neuron model. So I first generate these neurocrest cells that express the marker SOX10, and then I further differentiate them into sensory neuron, proprioceptive sensory neurons that express the marker for valbumin and also TUG1 and peripherin. Here in the bottom is our cardiomyocyte model. They start beating around day 8 or 9, as shown here in this video, and they also express the marker cardiac troponin T, shown here in pink. So using these sensory neurons and cardiomyocytes from the same patient and isogenic control, I am conducting global proteomic studies to identify novel mitochondrial targets that are altered in FA patients. Another focus of my research is to target oxidative stress in FA by targeting this MYC16-MIRO1 mitochondrial pathway. So mitochondria are known as the powerhouse or power plant of the cells, as they are the primary site for energy production, and they are also critical for the energy balance of the cells and several other cellular processes. So this protein, known as MIRO1, here shown in purple, is an outer mitochondrial membrane protein that anchors the mitochondria to the microtubule. When this MIRO1 protein is removed from the surface of the mitochondria, that acts as a signal for those damaged mitochondria to be degraded by mitophagy. So removal of MIRO1 is a way that the cell ensures that it is keeping only the healthy and functional mitochondria, and it is getting rid of the damaged ones. So in the presence of high level of reactive oxygen species, such as in Parkinson's disease or in FA, this MIRO1 protein is stabilized on the surface of the mitochondria, which further causes mitophagy impairment and causes accumulation of damaged mitochondria in the cells that can lead to cell death. So we are targeting this MYC60-MERO1 pathway using a small molecule known as MERO-reducer-3 or MR3 that our lab has previously shown to have beneficial effects in Parkinson's disease model. So here on the right, you can see some of our proteomics data from sensory neurons that are treated with MR3. So the graph on the left shows the mitochondrial protein and the graph on the right shows the whole proteome. So for both the graphs, if you look at the top left quadrant, those are the proteins that are upregulated in FA, and MR3 is able to reduce the expression of those proteins. And if you look diagonally in the bottom right quadrant, those are the proteins downregulated in FA compared to the control, and MR3 is able to increase the expression of those proteins. So currently, I am validating some of the hits that we got from our proteomics analysis in sensory neurons, and also conducting a proteomics analysis in the cardiomyocytes for the same patient and isogenic control pair. We hope that these datasets will give us exciting insights into the mitochondrial functions in FA. Yeah, that's a brief overview of my research. If you have any questions, please reach out to me. My email is mentioned here, sujotisi@stanford.edu. I would like to thank my mentor, Dr. Shinan Wang, all the patients that donate their cells for FA research, our collaborator, Dr. Mirella Dottori's lab in Australia that shared the iPSCs with us, and of course, FARA for the support. Thank you so much for your time.
All right. That was incredible to hear. And it was just a reminder to me of how important it is for everyone to be able to do what they can to contribute their cells when possible, or just be a part of the research. And we all kind of come together to make things happen. So I'm excited to introduce this next researcher who's going to talk a little bit about crossing that blood-brain barrier that we talked about earlier today that is so important. He focused on designing a shuttle that can deliver gene therapies across that blood-brain barrier. With that, I'm going to turn it over to Chang Fang to talk about designing a shuttle to drive medicines to the brain. And I want to thank you for being here.
Let me hide this float first. Hi, everyone. My name is Changfan Lin, and I'm a postdoc from Viviana Gradinaru's lab at Caltech. Thanks for coming to hear my story that how I try to build this shuttle to drive medicines to the brain. So as Liz mentioned, that it's very challenging to cross the BBB to reach the brain. And so far, the direct injection kind of works, by which it has to open up the skull and then inject the medicine directly into the brain, which is kind of also invasive. That's why our lab looks into these blood vessels that supply nutrition to the brain, as you can see here in the red color. So if we look deeper into this blood vessel, we will find it forms a vascular highway which reaches deep in the brain and also reaches more than 80 billions of neuron cells in the brain. So if we can use this vascular highway, then we have a non-invasive method that we can inject. We can just inject the medicine into the bloodstream and let it travel through the blood flow and reach the brain, and cross the blood-brain barrier and reach the brain. However, this highway is blocked by the BBB. So a lot of medicines has trouble to cross, but fortunately the nature made AAV, which can cross this BBB by itself. And in our lab, we are trying to make better version of this AAV vector to cross a BBB better. So to make it better, I always remember one thing that my grandma tells me: if you don't understand how it works, how do you make it better? So we have to first understand how AAV can cross a BBB. And then we will leverage that understanding to make it better. So if we look deeper, we can see this is showing the AAV flowing in the bloodstream. So when it reaches the BBB, it will interact with the receptors, which is showing here on the BBB cells. So after they engage, it will get a chance to get internalized and also cross the BBB to get to the other side of this BBB. So for us to make better AAV, we want to have an AAV which will bind to the receptor very specifically and also strongly. And that way we can avoid side effect and also make it crossing better. So with that in the lab, we select a brain-enriched receptor and use that to guide the selection of AAV variants, which can bind to this receptor better. Don't be overwhelmed by this scientific data here. I will walk you through this complicated data set. So all the dots that I'm showing here are showing different AAV versions that has different binding to the receptor on the BBB. And the y-axis and the x-axis, you can just think them as a characterization of this binding, see how strong it is and how specific it is. So our aim, our goal is to find AAV in the top right window, which shows it has good performance when it's bind to the receptor. And after that, we picked a short list of AAVs to be individually tested to see if they really bind this receptor very well. As you can see here, they are highlighted. The negative control is in the gray color, dark gray color. As you can see, it's now in this window. And then we have 5 AAV variants, which can bind very specifically and strongly. And then after that, we are doing in vitro tests to make sure they cross BBB better. And also we test them in the brain mimics, which is in vitro model of the human brain. So as you can see, the green dots are delivered by the AAV compared to the control, the wild type, the natural AAV, AAV9, our engineer AAV has a better broad coverage of the whole brain and also more efficient at crossing the BBB. So now we are in the process of testing it in humanized mice model or in non-human primate to see if it works. And with that, I want to thank the support of the FARA and also the support of my PI, Viviana Gradinaru. So as you can see, I'm not building a shuttle that can drive on the road, but I'm trying to build a shuttle which will drive in your mind to give you the hope that we can one day cure FA disease in the future. And that's it. Thank you.
Thank you so much for that. And I would say that a lot of us are very interested, all of us are very interested in that shuttle. So let's see what kind of questions we might have.
Sure.
All right, so Knight's talk. Can you conceive of other barriers to get to deep cellular nuclei after we surmount the BBB? Also, you were lucky to have a genius grandmother. I love it. Thank you for that.
Let me try to understand the question better. So the question is, can I conceive other barrier to get to the deep cerebellar nuclei after we surmount the BBB? Yeah, so that's a very good question. So the first layer of this brain target is to cross that river, the BBB. And after that, we want to be able to get into the cerebellum, which controls the motor function. And that's a second layer. So we are doing it step by step. After we can cross the BBB more efficiently, we are trying to also work on optimizing the specificity of its destination in the brain. in FA situation, we are trying to target the cerebellum better. There is another layer. So we wanna have, we will understand the receptor on the cerebellum cells as well. And as a second step, and try to have this dual targeting effect for the shuttle.
Thank you so much. Okay, so we have from Mary Scott. I remember meeting and talking with you at the Meet and Greet at the Philly Symposium. It's wonderful to see you again. Thank you for your continued work. I am so grateful. Oh, yes, I met you as well. Thank you for being there.
I really like that event, and I think I'm personally more motivated about working on my research because when I see the smile on the patient's face that they see the hope of cure FA, yeah, I'm very encouraged and motivated.
Thank you. That's so good to hear.
Maybe, Chanfang, do you want to talk about you had another project, so one other obstacle or challenge with AAV is the immune response that the body produces against these viruses. And so this is important for if you want to redose patients, or it's important when you first give the drug, if there's immune response that the drug, the AAV doesn't work as well. So can you talk about very briefly what are trying to do to overcome that limitation?
Yes. So the problem is that AAV is present in the nature by itself, and actually over 60% of people are actually exposed to AAV, and they have the immune system protecting us from AAV. So that's a challenge for us to use AAV for FA patients if they already have this strong immune system. So in our lab, we're trying two methods. One method is we try to understand how this immune system works with the AAV, and can we modify that part on the shuttle so it's not recognized by the immune system anymore. And another method we are trying is try to basically give a wrap outside this AAV. So without deeper understanding of how the immune system works, we can try to basically fake the AAV, so the immune system doesn't recognize, oh, it's a AAV anymore. So that's the two methods going on, and we are trying to get update good results in the future with all the FA people with FA concern.
Thank you so much for that. That continues to be fascinating. So there's one more question in our chat. I don't know if you can see it, but I'm going to read it. Do you think that this improved AAV will also target other parts of the nervous system, the DRG, for example, for a global action?
Yes. So it's a very good question. Can we target other tissues or other organs in the body? Yes, we can, because in this research, we are showcasing that if we understand how it works, we can make it better, as my grandma said. So a lot of people are working on to understand how the target, what the receptor is on the DRG, and what the receptor, for example, in the peripheral neuron system. So if we understand that, we can use the same workflow to engineer AAV for better targeting of these receptors to make it better. So fortunately, we are a teamwork that a lot of scientists are working on that aspect to understand the receptors. After that, we will follow up with our workflow to make better AAV for that specific receptor.
Thank you. Okay, are there any more questions before we move on to our next researcher? Thank you so much.
Okay.
This has been incredibly helpful. Sorry.
Yeah, and I just wanted to ask, do you also check if it's reach over, if it's not, even if it's not in your goal, do you check or verify that it doesn't target other parts of the body, so that you
Yes,
don't have kind
that's
of
a
side
very good
effects?
question. So to know that, that's an open question. If we test it in humanized mice, basically this is a mice which has a human receptor expressing in their BBB. So can we trust that data of biodistribution to be sure that it will mimic how it will distribute in the human? That's an open question actually for everyone, because for some research we found if you have AV works in mice, that it actually doesn't work in human because the biological mechanism is different in mice and humans. So we will check if it will go to other organs in the humanized mice, but we don't know if we can use that to see the biodistribution in human as well.
Well, thank you so much for being here and answering all these questions. I think we're going to move on to our last researcher. Our next researcher is going to be discussing research on a gene editing approach for FA using bone marrow stem cells. Welcome, Anusha. Can't wait to hear more about blood stem cells and gene therapy for FA.
Thank you very much for that, Elizabeth. I'm just going to share my slide. And first of all, I would like to thank FARA for giving me this opportunity to present here. And I would also like to thank again FARA for nominating me for an award in conjunction with Uplifting Athletes, which is also a rare disease organization. And both of them together are funding part of the work that we are doing here. I am a project scientist at Dr. Stephanie Cherky's lab in UCST. As a lab, we are working on developing blood stem cell and gene therapy for rare diseases. We have previously done a phase 1/2 clinical trial on another rare disease called cystinosis, and Friedreich's ataxia was the next study that, from the lessons that we learned on cystinosis, that we were able to develop. So I'm going to walk you through the story for the last few years on FA, and the story goes from left to right. So initially, as a proof of concept to say whether blood cells can help treat Friedreich's ataxia, what we do is we isolate hematopoietic stem cells. Hematopoietic stem cells are baby cells that are present in bone marrow, and these help produce all of the blood cell types that are present, and so they are part of the immune system that helps protect your body against foreign invaders. So what we wanted to do was see if we are able to make these blood stem cells healthier. Are we able to treat Friedreich's ataxia? So that was our initial study, where we used a fluorescently tagged blood stem cell from a healthy mouse and transplanted it into a Friedreich's ataxia mouse. And what we saw was very, very surprising, is that these stem cells had the capacity to go to all of the organs that are affected by Friedreich's ataxia, particularly the spinal cord, the heart, the brain, and the muscle. So all of those organs that have been reported to get affected by Friedreich's ataxia was able to accept these blood-derived stem cells to sit down there. And what we saw was that we saw an increase in frataxin expression in all of these organs. And this was done by all of these cells that engrafted into the organs, where they formed connection tubes between the healthy cells and the diseased cells. And these connection tubes helped transfer frataxin from a healthy cell into these diseased cells. And that helped cause a lot of rescue that we were able to see in both the neurobehaviors that the FA mouse was exhibiting, the cardiac complications that the FA mouse was exhibiting. So at this point, we had a vehicle that was able to treat the disease. And now we wanted to develop a method where we can use patients' own cells, blood cells, to treat the disease. So that led us to developing a gene therapy where we use CRISPR-Cas9, which is a popular method to edit your DNA. So with our approach, what we did was we developed a method to cut off the GAA expansion from the frataxin gene in the blood cells that were derived from FA patient volunteers, and we saw that when we were able to excise these mutation, these cells were now able to produce increased frataxin levels. Now, combining these two studies, which is what we have here, is our current study where we are trying to see if we use the
Oh, I think we lost Anusha. Let's see if she can come back.
While we're waiting, it's so cool to hear CRISPR being talked about, especially when it comes to FA. I know that CRISPR is something I've heard on podcasts, I've read about, but really to hear it being potentially put towards FA is really cool.
Yeah, it's a very powerful methodology, and there was just not that long ago the first therapeutic that was approved using CRISPR. So there is a path there to, for it to be used as a therapy.
While we're waiting, and hopefully she comes back to us soon, is someone comfortable kind of explaining what CRISPR is for those of us who really sometimes struggle to understand?
Sure. You want me to take this? It's like we always compare it to a molecular scissors, right? It's a protein that the bacteria actually is a bacteria protein developed, during evolution to defend themselves against viruses. And so when they encounter the bacteria encounters a virus, they save a piece of the DNA of the virus as a reminder. Next time I'm gonna see this piece of DNA, I need to react against it. And that's what the CRISPR protein does. It cuts that piece of DNA. And so, from the first discovery, as a sort of an immune response to viruses, it was kind of redesigned to target the genome, the DNA in human cells. And so that's where the application, for editing any part, because it can target any parts of the genome. It can be programmed to recognize specific parts of the genome, specific sequences of DNA. Then it can be used to, cut, or now there's other better versions of it that can be used not only to cut the DNA, but to replace pieces of DNA or to change just one single letter of the DNA code. So now the applications are really expanding from the initial, molecule that was the initial protein that was just cutting the DNA and making, this deletions in the DNA. I don't see Anusha coming back, unfortunately. So I'm afraid we'll have to stop here.
That was, and I'm gonna echo what Mary just threw into the chat, is that she loved the project and presentation so far. And so yeah, maybe we can find a way to grab that recording from her at some point and share it.
Yeah.
Because I was kind of on the edge of my seat there.
All right. Well, sorry about the technical problems. This is our lives today, nowadays. We can't do it without technology. So I just want to thank to all the speakers today. That everybody did such a wonderful job. And I hope you're all excited by all the great science that you heard today, and that FARA funds, the speakers of today's and the speakers from 2 weeks ago. And if you didn't have a chance to listen to their talks, they are on YouTube, as I said. And thank you for the moderators for helping us with question, Beth and Veronique. And thank you all for listening. Thank you for raising the funds that support this research, for participating in the research, by, you heard, donating your blood, your skins, how, you know, this researcher use all these donations that you make, the skins, the blood, the muscles, and also for participating in clinical trials and in clinical studies. And have a good day, everyone. Oh, there she is.
Oh, welcome back. We're so glad you're here.
Yes, very sorry about that. I'm just
Maybe
gonna
we
get back.
can take 1 or 2 questions on what you presented, if there are.
Could I get a minute to just wrap it up?
Sure, go ahead.
Thank you. Yes. So as I was saying, yeah, we have a vehicle and we have a gene therapy method. So what we're doing right now is trying to see if we apply both of these methods into a FA mouse. Are we able to see the same efficacy? Are we able to see the safety of this approach when we reach patients? So what we work with right now is a Friedreich's ataxia mouse model that has about 800 GAA repeats. And so it makes sure that it applies to a broad range of patients that we can treat. And we transplant these mice with gene-edited cells. These gene editing is done outside of the body, where we collect patients' bone marrow stem cells from the blood and then do CRISPR-Cas9 correction and then inject it into the mouse. So what we see is that when we did this gene correction, these cells were now able to localize to all of the organs that are affected by Friedreich's ataxia, which is what you see in the top graph. And particularly in the spinal cord, the brain, the cerebellum, and in the heart. And what we see down here are images of how the cerebellum would look like in a Friedreich's ataxia mouse, where we have very significant loss of the neuronal numbers. We are looking at this purple stain over there. And when we did the treatment to these mice, where they received the gene-edited cells, we were able to retain a lot of the neuron numbers that were gradually getting lost in a FA mouse model. And what we also see in the heart, which is where the cardiomyopathy and the hypertrophy develops and leads to death in these patients, is that the myofibers were organized so much better than what FA mouse had when you treated it. And we also had healthier looking mitochondria, which are supposed to be organized in parallel fibers between the myofibers. So ultimately, with the end of the study, where we are trying to see is when we do the transplantation, are we able to treat Friedreich's ataxia in the mouse model and eventually develop it into a clinical trial where, again, we would collect the patient's bone marrow stem cells, do the CRISPR-Cas9 correction outside of the body, make sure that the cells are healthy enough to be given back to the patients, and it will be a one-time therapy for Friedreich's ataxia. And that's the end of my slides. So I will have if you have any questions, I'm happy to take them now.
Thank you so much for coming back. This is absolutely fascinating. And I will tell you, as a mom, seeing that you focused on looking at the cerebellum and then the heart cells, we know that those are 2 incredibly important pieces to be mindful of on these bodies that have FA. Okay, so our first question, hi Chandra, is from, it says, what age stage of FA were these mice at transplantation?
So we transplant these mice at 2 months of age, and that roughly translates to about 7 to 10 years of age in patients. And our focus was on this particular age so that we can try to interfere either when they are diagnosed early or not so severe. That way we can assess whether there is a difference in the treatment.
Thank you. Okay, here's another one. Is there a way to increase homing to and differentiation in the CNS?
What we do as a therapy is, again, for hematopoietic stem cell transplants, the way of doing that has been established a long time for a lot of diseases, including neurological diseases. And so what we have right now is untouched system where you do a normal process and it seems to work. And our target is to reach all of the organs that get affected, including the brain. So with what we do right now, we were able to reach to the brain and the cerebellum. Would we like to have it better in the brain? Yes, we would, but that's not something we have started yet. But it's as a study we are trying to explore if there are other ways of conditioning the mice where we can have them come better to the brain and at the same time reach all of the other organs that are in the periphery. So it would depend on conditioning to draw these cells, and that's something we are working on in parallel.
Thank you. Okay, are there any other questions? We hear from Barbara Tate. Well done, speakers and moderators. Thank you. Thanks for being here and for all your support. Okay.
All right. All right. Thank you for coming back, Anusha. And we're
Thank you.
wrapping it up.
Thank you.
And thank you.
you.
Thank you, everybody. Thank you, speakers and moderators. And we'll see you next year.
Thank you. Bye-bye.
Thank you.